Thank you very much, Professor Poltzeff and Professor Sukov, for inviting me here to tell you what we've been doing, and for the opportunity to see this wonderful city here that you have, St. Petersburg. Well, I'm going to talk a little bit about results, because I've been noticing the lack of the clinical focus so far today, and yet the conference is called Molecular Basis of Clinical Medicine. So let's talk a little bit about the clinical side of things. Well, we started a worldwide collaborative study back in about 2004, and by 2008 some of our international collaborators were starting to publish their results. And at the International Congress on Autoimmunity in Porto, in Portugal, in 2008, Dr. Greg Blaney from Canada reported on a few very interesting case studies. The first one was a patient with progressive multiple sclerosis, an EDSS around 8, and in that case the disease had reversed and the patient was back to about 7 and was mobile again. A patient with a diagnosis chronic fatigue syndrome, myalgic encephalomyelitis, with also positive ANA, and who was positive for Borrelia, doing well, and a patient with fibromyalgia and chronic fatigue syndrome, who was also doing well. But also, the very first of our study analyses was done. We did a retrospective on about 100 or so of the 1,000 totally enrolled at that particular point. And Captain Tom Perez, recently retired from 25 years at the US FDA, summarised those results. And the interesting thing is that with this particular therapy that these collaborators were using, diseases that, or diagnoses ranging from rheumatoid arthritis through Hashimoto's, psoriasis, and somewhere in the middle is multiple sclerosis, of course, which you mentioned earlier, right to myasthenia gravis and diabetes insipidus, were all responding in some favourable fashion to the therapy that was being used, an immunostimulative therapy, not immunosuppressive, immunostimulative. 2 years later, at the International Congress on Autoimmunity in Ljubljana, Dr. Greg Blaney chronicled 3 cases: ankylosing spondylitis, which responded very quickly, autoimmune arthritis with Raynaud comorbidity, rheumatoid arthritis, and aggressive rheumatoid arthritis. The thing that's very interesting in all of these cases is by this time the endpoint which the clinical collaborators were looking to achieve was return to work, return to the family, return to work. In other words, the disease reversal rather than just disease containment. By last year at the Autoimmunity Congress in Asia, Dr. Goetze-Pelke from Berlin gave some results of patients with sarcoidosis and multiple sclerosis who had recovered well, but in particular was focusing on the psychiatric comorbidities that had also recovered along with the inflammatory disease, pointing out that the psychiatric disease and neurological disease appeared to be very, very tightly coupled to the underlying systemic inflammatory processes. She gave 2 case histories, once again with the endpoint of return to work. And just a few weeks ago at the Autoimmunity Congress in Granada in Spain, Inga Lindseth from Norway gave a case series demonstrating resolution of CFSME using this drug that they had been using, olmesartan, once again with an endpoint of return to work. He had people with I recall about 2 to 6 years of therapy, and around 20 out of 60 were able to return to work. At the start of the study, only 2 were in gainful employment. So it's not all that unusual for such a wide range of diseases, such a wide range of diagnoses to be affected by one therapy. For example, TNF therapy and prednisone, the immunosuppressive therapies, have been used for decades. But in this case, it was a diametrically opposite therapy that was in use. It's an immunostimulative therapy. So how could this possibly work? At the molecular level, what is happening here? And the clue lies in molecular mimicry. You know, just a decade ago, the human genome was decoded, fully decoded, and there's been a burst of discovery since that time. But, you know, the biggest discoveries are not the ones that you've been reading about in your paper, your newspapers, or even, in fact, hearing about at the major conferences. Because the biggest discovery is the extension of the human genome to the understanding of the microbiota that is Homo sapiens. Homo sapiens is a superorganism. Each one of us is carrying around thousands of species and subspecies of other organisms. It's complex how to describe them. Let me call them other organisms. The human microbiome. They're in all our tissues, they're in our blood. Any sample of blood from a live human being, run that through a sampler, through a sequencer, or through PCR, you will find the organisms. It's absolutely clear. And several years ago, 2007, the National Institutes of Health started the Human Microbiome Project to try and find out exactly where these microbes, what these microbes were doing and where they were in our body. And those of you that read the Los Angeles Times or the New York Times of yesterday would have seen front page stories on the results of the HMP project, because it's now finished. Late last year it finished. And the Human Microbiome Project identified millions of genes belonging to thousands of species of microbes which live in and on the human body. 25,000 human genes, millions of microbial genes. Over 3 million was the estimate that was given by NIH yesterday. Plus all the viral genes and the fungal genes as well. And in fact, our microbes make us genetically unique. Everybody has a different, slightly different family of microbes. Our genes might be fairly similar. We may have some SNP differences. I'll talk about them later. But basically, our microbes are very different. No matter which part of the body we sample, the microbes in each of us are different. And more importantly, those microbes change from week to week and month to month. Our microbes change. The balance is continuously changing. And at some points in our lifestyle, some of us will get a balance that causes dysfunction of the human genome and we become ill. Metagenomics is the study of those microbes. And the textbook was published last year, Metagenomics of the Human Body. The human body contains an ecosystem of microbes. And we wrote the chapter in there on autoimmunity. And the important thing is to note that subclinical chronic infection is now confirmed, and not just a concept. It's been debated for decades. You know, can microbes persist in the human body? Yes, they do persist. Thousands of species persist. And in fact, babies are born dirty. When you are born, you have a good complement of microbes. I was going to say a healthy complement, not necessarily. Some babies are born ill. But a good complement of microbes when you are born. And from that point on, you can't avoid accreting or gathering more microbes as you go through life. This is data from the Craig Venter Institute, and it shows organisms in air samples, DNA in air. And you can see in a hospital, of course, in San Diego Indoor Hospital, or a hospital indoors, that there is a large percentage of bacterial DNA in the air. That's probably no surprise in a hospital. But you go into a house and you find about the same percentage. About 85% of the air floating around in the house, of the DNA in the air floating around the house, is bacterial. And then there's some human content, fungi, insects, rodents, etc. If you take an outdoor sample, then the insects tend to be a very large relative proportion. And you can't avoid it in food either. This is a wonderful study that was done at MIT by Eric Alm's group, and what they did was a heat map showing the locations on the human body where antimicrobial resistant genes were present, where bacterial genes were present that generally were regarded as conferring antimicrobial resistance. And you can see a mirroring between the human genes that were found from the human body and those that were found in food from farm, agrarian food, soil, and then it goes quiet from that point on. But there is a huge correlation between the bacterial genes that we get from our food and the bacterial genes that collect in our body. And of course, in these last few decades of travel, international travel, to which we're all contributing, and the international transport of food, particularly, we're eating food, in many cases, US food comes from South America and from all over the world. It makes it much easier for the microbes which exist in the human body and indeed in the body of the farm animals to spread. But look, the really important microbes, the ones you read about in the New York Times yesterday, were the microbes that are in our gut. It's really easy to study microbes in our gut, and most of the early studies that have been done on the microbiome have focused on the GI tract. But the really important microbes are the ones that manage to get inside the nucleated cells and live within the cells, as the last speaker pointed out. The key is to get inside the cells. If you're a pathogen and you can get inside a cell and persist inside a cell, then you can do whatever you like. And that's been noticed before. This was a study that was done at Columbia University back some time ago by Wirostko's group. And what they did was stain for nuclear DNA in phagocytes, that is white cells, from patients with a number of chronic diseases, and they found in the cytoplasm of these phagocytes there were colonies of microbes. Different types of colonies, of course, different types of microbes. So the very phagocytes that were supposed to kill the bacterial pathogens were providing safe harbour for them. And this is a similar case. This one is from an infected lymphocyte from a juvenile rheumatoid arthritis patient. You can see a cluster of a vacuole which contains infected DNA or infected organisms in a vacuole type environment, and also long slivers as well. You can see them with an optical microscope. This was provided to us by one of our collaborators, and this is a monocyte which has just exploded as the cytoplasm got too tight, and these huge long filopedia have been thrown out by the microbes in that cytoplasm, in the infected cell. See if I can bring it back again. In the infected cell, the cytoplasm exploded. Nucleus is basically intact. It's the cytoplasm that gets infected. And then these long, thin tubules that contain the microbial DNA attempting to spread from cell to cell. So it was pretty obvious to me about a decade ago that I needed a different type of microscope. With the optical microscope, you could see some things. With the electron microscope, you could see other things. But neither of them allowed you to see the molecules. Neither of them allowed to see what was happening at the level of the molecules. And it's clear, as Professor Paulks have said at the start today, the molecules are where it's all happening. The molecular interactions are key to understanding disease. So what we put together was a computational microscope capable of looking at molecules within the cell and determining how the molecules interact and, in fact, how they exist at the cell, inside the cell. We're not the only people doing this. There are others in the world that have done it. But we did it a little bit differently, and we achieved some results. And, of course, the microscope is made of chemistry, physics and math. You basically emulating at any point where each atom in the molecule is. And of course, supercomputers. A lot of computing power. And that allowed us to do things like this. What I've done here is I've got the human angiotensin II receptor, the angiotensin II type 1 receptor. It's got a drug in the binding pocket, the drug olmesartan that we spoke about earlier, in the binding pocket. And over here I have the protein YDGG from the E. coli genome. This is an E. coli receptor, transmembrane receptor. I don't know what E. coli uses it for. Maybe it's in a molecular mimic. But anyway, if you look at the two receptors, you'll find the molecular structure is essentially identical. The molecular structure of the protein, folded protein from the E. coli bacterium and the human angiotensin II receptors are essentially identical. When these are mixed in the cytoplasm, it will cause all sorts of problems for the Golgi mechanisms and the other mechanisms that drive human metabolism. And it's interesting, the main difference is there is some difference in the specific amino acids, and the olmesartan drug docks in a slightly different position inside the receptor. Well, wait a minute, the drug docks inside a bacterial protein? Our drugs, the drugs we're taking, directly affect bacteria? That's an interesting observation in and of itself. But you know, bacteria also have metabolisms that are very similar to man. Now, the eukaryotes are special, but even the prokaryotes are somewhat similar because there is an evolution process that are linking them together. And if we take microbial metabolism and look in particular at the glucose metabolism, we find that E. coli gets its energy from glucose 6-phosphate, and it generates pyruvate at the bottom. But each of these arrows, 7 arrows if I remember, are specific intermediates in the glucose cycle for E. coli that are identical with the substrate metabolites in man. In other words, there are genes, which are listed here, there are genes in the E. coli genome that are identical in function, but slightly different in molecular composition, but identical in function to those that are in man, in Homo sapiens genome. Very, very similar, but slight differences. We've been hearing that so much from the laboratory people, saying, look, we're seeing dysfunction, we're seeing SNPs, we're seeing longer substitutions. Maybe we're seeing the bacteria. So in this particular case, molecular mimicry is not any specific attempt by the microbe to mimic a function. It's the microbe needs to live. It's got to turn glucose into energy, and it does it the same way that we do it. But you know, 2 speakers ago, we heard that the mouse is not a very good model. We now have a complete genome of the mouse, and in fact it is amazing how different the mouse is to man in terms of the way it actually works. So what we were able to tell from our molecular microscope working with Homo sapiens genes was that in Homo sapiens, and only Homo sapiens, not in the mice, not even in the higher primates, there is a key immune function that is weak. In Homo sapiens, there is one nuclear receptor which expresses the genes for a key toll-like receptor 2, as well as cathelicidin and beta-defensin antimicrobial peptides. And all of those, of course, are essential to intracellular innate immune defences. So in order to survive within cells, microbes clearly need to evolve to knock out that receptor. And that's exactly what they do. The microbes that are specialised to knock out that receptor are the typical ones that you've seen in chronic disease. EBV knocks it out up to a factor of 15 times in a lymphoblastoid cell line at 1.5 years, but also Mycobacterium tuberculosis, Borrelia burgdorferi, Chlamydia trachomatis, Aspergillus, HCV and CMV all specifically manage to suppress the innate immune system and survive by knocking down expression of and by the VDR nuclear receptor. But you know, the microbiome also messes with our human biology. They not only knock down the VDR, which definitely affects our human biology. VDR is a very important receptor. But they knock it down in millions, an imponderable quantity of other ways as well. And for example, a dysfunction X, which may be a disease symptom, can come with genes from bug A combining with genes from bug B and causing a dysfunction X. And also you can have multiple bugs with one bug or one microbe missing. For example, some microbes are antagonistic to each other. Strep and staph seem to get at each other's throats all the time. So you have both combinations of microbial components that will cause a human dysfunction, and then you also have missing components as well. And the genomes of the microbes accumulate gradually during life. It's genes from the accumulated metagenome at any point in life which determine the clinical dysfunction and the disease symptoms. And that's why you see the comorbidities. When I was first studying diabetes, 1978, many years ago, one of the things I noticed was that the patients that we were treating for diabetes also tended to have asthma, rheumatoid arthritis, hypertension, a number of comorbidities. And this particular chart came from Dr. Prohl's paper in Metagenomics of the... actually came from our chapter in the book, my colleague Dr. Prohl. But that was obtained from some studies that were done on comorbidity that are published in PubMed. Very interesting, the links between all these diagnoses. We like to think of disease as separate, but we can't. Now, something else we were able to do with our molecular microscope is see how the molecules interact. And this is actually the VDR receptor, and across this region of the receptor is the coactivator, which needs to be bound in order to produce the heterodimer and translate genes. And we were able to show with the drug that we were using in the human receptor that the active atoms here were ready to bind to the coactivator. And we were also able to show that in the rat that was not the case. In the rat the drug does not work. What happens is this. You've got the VDR nuclear receptor, which is very important. It transcribes at least 1,000 genes. Probably as much as a quarter of the total genome is affected. Expression is affected by VDR. And normally there is a steroid calcitriol that activates the VDR. But when the microbes come along, the microbes have toxins. For example, gliotoxin is one of the toxins. Many ways that they can knock out the functions of the VDR. The microbes knock out the activation of the VDR, and that stops the VDR from producing the antimicrobials that would otherwise kill the intracellular microbes. And this drug that we found, olmesartan, is capable of reactivating the VDR, provided the dosing is changed. You see a normal dosing of olmesartan is a single peak and then it trails away through the rest of the day. When we retarget the drug for multiple dosing during the day, you can see we keep a basal level in the bloodstream, and that allows the drug to have totally different effects from when it's given as a blood pressure drug, because it was developed as a blood pressure drug. Because we had a drug that's already approved by the FDA that could be prescribed with different dosing off-label, we were able to go straight to patient-important outcomes. We weren't distracted by worrying about the mouse and how the mouse behaved and the differences between mouse and man. We were able to go straight to the patient-important outcomes with our clinical collaborators. And what we found was this, that in order to help people with chronic disease, huge wide range of chronic disease, about 120 diagnoses in our cohort. You have to not only deal with the inflammation, you have to also deal with the molecular mimicry. The inflammation gives rise to cytokine storm, autoantibodies, nitric oxide, ROS, urea, etc. But then the molecular mimicry provides the dysfunction, the interactome damage, which leads to the body dysfunction, the cortisol dysfunction, the thyroid dysfunction. You have to deal with both of those. Just attacking the inflammation does not prevent the spread of the disease, nor does it exacerbate all of the disease symptoms. In a way, you can think of these persistent intraphagocytic infections, the intracellular infections, as something like white ants. You know, with white ants in a building, there's no obvious sign of damage until the whole structure starts to crumble. And that's the stage we are at the moment in medicine. And we've got to move towards a stage of predictive medicine, where we can say, look, there seem to be white ants in this structure. The microbiome gives us the ability to do that. Of course, there's always a problem. And in this case, the problem is immunopathology, which means that when the immune system starts recognising the microbes within the cells and killing the microbes within the cells, many of the cells die as well. Plus the immune system is more active. You have a surge in the cytokine storm, you have a surge in the immune functions, and that is what is termed immunopathology. So the pathogen is exerting direct damage onto the host. The host-resistant mechanisms exert damage back onto the pathogen. But there is some of this damage caused by the immune response that gets back and hurts the host a bit too. And this is immune pathology. We covered it in detail in our paper, a recent paper there. I won't spend any more time because we're tight on time on that. But immunopathology has to be dealt with, and it has taken us a decade to figure out how to manage immunopathology in order to induce recovery. So predictive and preventative medicine. Now that we have a working model for autoimmune disease processes, it becomes practical and sensible to start treating patients early. For example, as soon as we see autoantibodies, typically with the new tests that are available, autoantibodies can be found years before any symptomatic dysfunction occurs in an individual. Most of us are carrying around autoantibodies. The Red Cross blood supply has autoantibodies in it. So when people first start to exhibit with autoantibodies, maybe that's the point at which we need to say, look, you shouldn't have those. There's a dysfunction. Let's start looking at your immune system. Measure the markers of the immune system. The VDR effectiveness is one of the markers we can use. There are some CYP 24, for example, which is characteristic of VDR that has been measured clinically as a VDR indicator. But at that point, we can initiate therapy, because olmesartan, the drug that overcomes the effects of the microbes, is one of the safest drugs in the US formulary. I hate saying a drug is safe, especially when I'm espousing its use. However, it is a very, very safe drug. The FDA has set no unsafe dosage for this drug. There's no known unsafe dosage for it. And it's FDA approved to be taken for a lifelong dysfunction, which is high blood pressure. Our off-label retargeting does change the safety profile a little because of immunopathology, but the earlier that a patient begins therapy, the less is the amount of immunopathology they exhibit. In fact, healthy people exhibit no immunopathology at all. And that's very important, because the drug would never have been approved if the cohort were exhibiting immunopathology at the same time as a lowered blood pressure. So the earlier we get the patients, the much easier it is to stop the disease progressing in the long term. So maybe we have an opportunity here for predictive and preventive medicine. Now I want to talk something else as well. There's a huge impact of the microbiome on the work that each one of us does. Because when you take a sample from a human being, whether it be blood, or whether it be tissue, or whether it be cells, that sample contains microbes. It contains microbial DNA, which you can find with PCR, and it contains, or with a sequencing approach. And you have to be very careful when you're doing a study of SNPs or mutations that the DNA that you're actually looking at is human DNA, that all of the microbial DNA has been stripped out. And this was realised some time ago by the US government, of course, after the anthrax attacks in America, and they put a lot of money into searching through DNA databases to find which of the microbial genes and which of the human genes. And if you look at that particular URL from Argonne National Labs, you'll find that any studies that you've done of human body where you haven't sorted out the microbes, they will help you do it either by providing the software or providing the computing power. There are a number of other very important papers here that I've listed for those of you that are doing genetic testing and genetic analysis to help you better analyse your sample and make sure that what you are thinking is a SNP is actually a SNP and not just a piece of microbial receptor that looks awfully the same as the human receptor. Because what happens is the software that has been provided you for your genome sequencing is very faulty. It only needs 1 or 2 base pair errors, and it goes totally wrong as it forces the RNA up into the human genome that it has in its data bank. Because when you feed in an RNA sample into one of the sequencing machines, it's trying to put that RNA up into the human genome that it has remembered in its memory, the standardised human genomes we've had for a decade. And as we can see from these graphs here, which came from actually Barry Merriman's paper, he checked for 6 of the major pieces of software that are used by all the commercial manufacturers, and he found that just a one position deletion was enough to cause 10-15% errors in the mapped reads. And the same with an insertion. Just a 1 position insertion was in some of the software enough to cause more than 10% poor assembly of that RNA. It's very, very important. We have to remember that every sample you're working from with a human being has got microbes in it. And if you look at the DNA and the RNA, you'll find them. So here we are, take-home points. There's a lot of them, I'm sorry. First take-home point is Homo sapiens is a superorganism. It's a symbiotic metagenome. When the metagenome becomes unbalanced, then disease ensues. Intraphagocytic microbiota persist by suppressing innate immunity, knocking down the VDR nuclear receptor. And all the antibodies that we see, the autoantibodies that we see, are of course a cascade. Once the innate immune system is compromised, then the cytokine release causes the adaptive immune system to say, wow, we better start working extra hard and try and deal with this problem that the innate immunity has. So it's the innate immune system that's key. Olmesartan can reactivate the VDR and reactivate immune function if it's used properly. But you have to be very careful because many people will get very, very ill. Nothing like having a call from a hospital that it was in the middle of the night, I think about 10 years ago, and one of the subjects had a heart rate of 10 with occasionally missing beats. You don't want that sort of thing. That is too much immunopathology. You have to control the immunopathology. And the temporary worsening of disease is measurable. Usually the patients feel better right away, but you can measure the temporary worsening of disease. Read our paper for that before the healing becomes dominant. And before commencing a genetic study, always remove the microbial RNA. And for those of you that have already done a study, you can take your raw data and feed it back through Argonne Labs, and they will take the microbial RNA out of that for you, so you'll have a cleaner sample to look at again. Disease is complex and not simple. It's pretty obvious. I think just about everyone has said that here. But the reason for that is because the interactome, the potential for interactions between the millions of microbial genes and the 25,000 human genes is semi-infinite. Semi-infinite is really the only way that we can quantify it at this point. So thank you very much, Mr. Chairman, and once again thanks to the organising committee.
Я прошу извиниться.